Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
So disaccharides are formed by condensation reaction between two monosaccharides. So some examples might be maltose, sucrose or lactose. Polysaccharides are formed from a multitude of glucose subunits. So glycogen, for instance, and starch are formed from the condensation of alpha glucose molecules. Cellulose is formed from the condensation of beta glucose molecules. So we can use a Benedict's solution to go and carry out a chemical test to look for both reducing sugars and non-reducing sugars. Or if we're looking for the presence of starch, we can use a iodine solution that turns black to look at that. So the basic structure and functions of glycogen, starch and cellulose are quite different. So their relationship very much is dependent on their structure and the job that they do within animal cells and plant cells. So disaccharides are made, like we've said, when monosaccharide subunits are combined. So the different or similar monosaccharide molecules are attached via a glycosidic bond, and that glycosidic bond forms a disaccharide to begin with. So when we form a glycosidic bond, water is released because it is a condensation reaction. Right, also it can be called a dehydration reaction, but generally we call it a condensation reaction. So a glycosidic bond or a glycosidic bridge, so the oxygen acts a little like a bridge that goes and connects the different carbons of two different monosaccharide molecules. Like we said, we call that a glycosidic bond. So we're going to look at starch. Right, starch is primarily found in animals and is a store of energy and is also important for lots of animals' diets. So the two forms of starch that we get are amylose and amylopectin. Right, amylose is broken down by the enzyme amylase, for instance. So we go and we make it up of alpha glucose monomers. Right, so amylose makes up around 20 to 30% of starch. Right, it's unbranched. Right, that unbranched part is quite important. Right, and it forms a linear change of glucose molecules. So these glucose molecules are linked by a 1,4 glycosidic bond. Right, and we can see there is a structure. So we can see there is where the glycosidic bond is forming. Amylopectin, right, shows branching right after about each 24 to 30 glucose subunits. So it's more branched than amylose. So each branch contains about the same number of glucose subunits. So the glucose subunits in a single branch, again, joined by an alpha 1,4 glycosidic bond, right? And we can see there, there's one branch, there is a second branch, and we're forming it again via a glycosidic bond just there. Right? That's where your normal glycosidic bond would be. Right, glycogen, however, so glycogen is a multi-branched polysaccharide of glucose that serves as an energy store in animals. So glycogen is made primarily in cells in the liver, right, and the muscles where we need to go and store glucose to be used later. So, like we said, so glycogen is functioning as a long-term energy store. Right, muscle glycogen is converted into glucose by the muscle cells, and the liver glycogen converts into glucose to be used throughout the body that is then released into the bloodstream. So, like we said, glycogen is a glucose polymer that functions as an energy store. One of the key things, right, glycogen, because you can see here, it's very, very multi-branch, so it's got lots of free ends, can be quickly metabolized to meet the sudden need for glucose that you might need if the rate of respiration increases. So, in the liver, glycogen is produced, and you can see that it's produced really quickly because lots of enzymes, in fact it's made and broken down really quickly because lots of enzymes can act on all of these branched ends. Right, if we just go and look at a quick question. Right, so glucose and glycogen and amylose are carbohydrates. So glycogen and amylose are used as an energy store. Right, glycogen, like I said, is found in animals, and amylose is found in plants. So we're looking at describing how, right, so the key thing there is describe how the structure of glycogen allows it to perform its functions and explain the advantages of using glycogen as an energy store. Right, so the point is, you're not just describing it or you're not just explaining it, you've got to kind of do both. So you need to talk about the fact that it contains alpha glucose, which is a respiratory substrate. You need to talk about the fact that it can be broken down easily or hydrolyzed by enzymes. You could talk about the fact that it's making a large molecule that has lots of long chains. Quite importantly, it's insoluble, so that's why it can be stored. So it does not affect the water potential of the blood, for instance. It's relatively compact, right? So it's dense, so it can store a large amount of energy in a relatively small volume. You can talk about the fact that it forms 1,6 glycosidic bonds, so it's branched. Again, we can talk about the fact that it can be broken down at multiple sites by enzyme activity, which means that it can be broken down quickly. And because it can be released quickly, it can go and be used to generate more ATP that's required for a range of cellular activities.
So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.